When two tall pea plants produce a dwarf offspring, they're following a specific mathematical probability hidden within their genetic code. That logic was first mapped out by Gregor Mendel. Through his experiments, he built the baseline rules for passing traits to the next generation. To observe these rules, Mendel needed a test subject with easily identifiable contrasting physical traits. He chose the pea plant. A plant was either definitively tall or dwarf, with no middle ground. Mendel observed that crossing a pure tall plant with a pure dwarf plant resulted in an F1 generation that was entirely tall. To explain why the dwarf trait vanished, he proposed the law of dominance. When a plant inherits both a dominant allele, T, and a recessive allele, T, the dominant trait is the only one expressed. But that hidden dwarf allele does not vanish. The law of segregation dictates that when this plant forms gametes, that allele pair must physically split. Each gamete gets exactly one allele, guaranteeing they never mix. If we take two of these heterozygous TT plants and cross them, we map the probabilities using a Punnett square. The separate gametes recombine, giving us three combinations that include a dominant T and exactly one combination of two recessive Ts. This mathematically resolves the appearance of dwarf offspring in the F2 generation. 3 out of 4 offspring will look tall, giving us a 3-to-1 phenotypic ratio. Underneath, the genetic code breaks down into a 1-to-2-to-1 genotypic ratio, 1 TT, 2 TTs, and 1 TT. These ratios, 3-to-1 for physical traits and 1-to-2-to-1 for genetics, form the baseline of complete dominance. This predictable outcome serves as the standard against which all other inheritance patterns are measured. The law of dominance works perfectly until neither allele is strong enough to win out. To test this logic, pause the video and map out a cross between two pink snapdragons. Determine the phenotypic ratio of their offspring. The resulting square gives you 1 RR, 2 RRRs, and 1 RR. Unlike complete dominance, where heterozygous offspring look like the dominant parent, here the heterozygous genotype always displays the blended intermediate trait. A second variation occurs when both alleles are fully and equally expressed at the same time. This is observed in the human ABO blood group. If a person inherits an A allele from one parent and a B allele from the other, their red blood cells display both A antigens and B antigens concurrently. These alleles do not blend to create a new hybrid antigen. Instead, both are present and active at full strength. This is codominance, a scenario of shared genetic power where the phenotype fully exhibits both traits at once. These ratios allow for reverse engineering of inheritance patterns. An F2 generation with a 3-to-1 phenotypic ratio identifies the mechanism as complete dominance. When an F2 generation displays a 1-to-2-to-1 phenotypic ratio, the distinction lies in how the traits are expressed. Descriptive terms like intermediate signify incomplete dominance, while both, or equally expressed, indicate codominance. This comparison table maps out all three inheritance types side by side. It traces how the rules, the classic examples, and those critical phenotypic ratios change as the mechanism shifts from standard complete dominance into the two major exceptions. Grab a screenshot of this table. Identifying where these patterns differ is the most direct way to ensure accuracy on genetics ratio questions.